wrap up our last lecture and discuss these last 3 learning objectives. So remember the adrenal medulla. I had mentioned in my previous lecture how it was an exception to the whole, you have a preganglionic neuron and then that synapses in a ganglion with a post ganglionic neuron, right? So a preganglionic neuron to a postganglionic neuron. But with the adrenal medulla, there was no typical postganglionic neuron, and that's because the adrenal medulla itself the cells of the adrenal medulla are modified postganglionic neurons, essentially, right? So here we have our preganglionic neuron exiting the spinal cord, coming down and synapsing directly on chromaffin cells of the adrenal medulla. Remember, adrenal glands, they're pyramidal shaped glands that sit right on top of the kidneys. They have an outer cortex area which secretes different hormones, which we're not going to talk about in this lecture. And then the medulla, which these medullary cells are those modified postganglionic neurons. So upon stimulation, these chromaffin cells will directly release epinephrine and norepinephrine directly into the bloodstream. So this medulla is highly vascularized, and these hormones they get dumped into the bloodstream, and then that allows them to be circulated among the entire body, right? So these hormones will go and affect essentially all organ systems in the body. And a really important part of this is like the fact that so although epinephrine and norepinephrine from the adrenal medulla will have essentially the same effect on organs that the direct sympathetic stimulation will have, these hormones, they stick around in the blood a lot longer than they will stick around in synapses between effector cells, right? So this way of sympathetic stimulation allows for the effects to be more prolonged because they don't get cleared from the blood as easily. Now, why have direct sympathetic stimulation as well as adrenal stimulation and release of epinephrine and norepinephrine into the blood? Why have these two systems? It allows for coordinated, like synergistic effects between having direct stimulation as sympathetic stimulation and adrenal gland secretion. It also allows for like backup, right? So think about the sympathetic nervous system and fight or flight, being able to respond to stressful situations in terms of survival as humans, right? That system's going to be vitally important to survival. So it's good to have like these two mechanisms so that they can work synergistically when needed, but also if one fails, there's backup, right? So that's the key point or the big picture concept from this slide that I want you to know is that it allows them to work together, but also if one fails or if one's not working properly, there's backup. And then as far as the functions, let's see, the functions of the adrenal medullary hormones, epinephrine and norepinephrine, these are going to fall in line just like the functions of the sympathetic nervous system, which we talked about in the last video for this lecture. So you should be able to, if you know the functions of the direct sympathetic stimulation, you'll know the functions of the adrenal medulla hormones, epinephrine and norepinephrine. So be familiar with what types of physiological responses would happen as a result of epinephrine and norepinephrine being released. And so if you know the fight or flight responses, you'll be able to recognize that on a multiple choice question. The only thing that from this slide that would be, I guess, new or a new thing to be aware of, big picture concept from this is the fact that out of norepinephrine and epinephrine, epinephrine will have a much greater effect on cardiac stimulation. Okay? And epinephrine is released from the adrenal medulla, whereas norepinephrine not only released from the adrenal medulla, norepinephrine is also released at those postganglionic synapses with effector tissues. Okay, so epinephrine, greater effect on cardiac tissue. Now the concept of sympathetic and parasympathetic tone, what this slide is talking about is the fact that there are some organ systems that may have like a greater, might be under the control of the sympathetic or the parasympathetic system primarily or only. Um, so let's see. For the most part, the parasympathetic and your sympathetic nervous systems, they're working all of the time. So it's a very simplistic view to think that sympathetic is only fight or flight and parasympathetic is only rest and digest. They're working together all of the time, right? But that one might predominate over the other during certain events, right? Day to day. But then there are some systems which will have primarily or only sympathetic tone or parasympathetic tone, or be under the constant influence of one or the other. I hope that makes sense. Email me if it doesn't. So this slide is giving an example of blood vessels, so arterioles, which are under the control of the sympathetic nervous system and constantly have a sympathetic tone to them and are not innervated by the parasympathetic nervous system. All right, so for instance, sympathetic tone normally keeps all of the systemic arterioles constricted to about 1/2 their maximum diameter. So systemic arterioles are being, if you're just existing and you're at rest, these are systemic arterioles they're somewhat constricted because they're under the influence of sympathetic tone, right? And then what will determine them constricting further or dilating will be whether or not we increase sympathetic stimulation above normal, okay? And if we increase sympathetic stimulation, that means it's going to cause constriction, and we're going to decrease the diameter of these systemic arterioles, or we can decrease sympathetic stimulation below normal, and that results in dilation. So in this case, there's no parasympathetics at all coming into play. It's just whether or not sympathetic tone increases above normal or decreases above normal, all right? And then an example of an organ system that would be under a parasympathetic tone primarily would be like the GI tract, okay? The only thing I want you to know from this slide is the fact that the sympathetic nervous system, you can have a mass discharge of sympathetics, and that would be in a case of where you really have a fight or flight situation, you can have a mass discharge, but don't stress about this slide. And you would have all of the same things happen that we discussed or that you needed to know about the functions of the sympathetics. It just can be a really large mass discharge or output if needed. All right, so let's address these last two learning objectives with cerebral blood flow and CSF. All right, so cerebral blood flow, there are two major supplies, so 4 large arteries that are going to provide the majority of the blood to the brain, and you have 2 carotid arteries and 2 vertebral arteries. And what happens is that these large arterial blood supplies will form a structure called the circle of Willis. And the only thing that you need to know about this is the fact that this circle of Willis is going to provide collateral blood flow, so that if there's arterial disease or blockage of one of these major large arteries, that hopefully we can still get blood flow to the entire brain and prevent as much ischemia as possible. So the fact that these large arterioles form the circle of Willis provides potential collateral blood flow if one of them were to get blocked, okay? Now, normal blood flow, I'm not going to ask you to remember values here. I just want you to get an idea of how important cerebral blood flow is. Normal blood flow under resting condition to the brain of an adult individual is 750 to 900 mils per minute. Now, to give you something to compare that to, right, at rest, skeletal muscle blood flow may be 1 to 4 milliliters per minute. So we have, and if you think about how much skeletal muscle there is in the body compared to your brain, that's a really large difference. Now, under, exercise conditions, we can increase that blood flow to skeletal muscle, but at rest, it's only 1 to 4 mils per minute, whereas at rest for the brain, 750 to 900 mils per minute, right? So lots of blood going to the brain, and the brain. So although the brain comprises only 2% of your body weight, it receives 15% of resting cardiac output. To give you something to compare that to, at rest, skeletal muscle accounts for 20% of the cardiac output. And there's a lot more skeletal muscle than there is brain tissue, right? So almost as much as skeletal muscle, but the brain only comprises 2% of body weight. Main thing from this slide that I want you to know or be aware of is the fact that, what's the function of the circle of Willis? Regulation of cerebral blood flow, so maintaining blood flow to brain tissue is really, really important. And if you remember back from my first lecture when we talked about smooth muscle and the different types of chemical factors that can affect smooth muscle constriction or dilation or relaxation rather, these factors might look familiar. Carbon dioxide concentration, hydrogen ion concentration, oxygen concentration, and then other substances that can be released by supportive neuroglial cells like astrocytes can all affect blood flow. So, potentially increasing blood flow to the cerebral cortex if necessary. All this graph, the only thing I want you to, take home from this slide is if we look over here on this graph, so this is arterial PCO2, so carbon dioxide pressure in the blood, and then cerebral blood flow. And if we increase PCO2, so the amount of CO2 present in the blood, we're going to increase cerebral blood flow. And that if you think about it, it's kind of intuitive. If we're increasing CO2, that means we're increasing the amount of hydrogen ions present. We're making it a more acidic environment. We want to flush all of that out and bring more oxygen in. So we need to increase cerebral blood flow in that instance, right? Because any imbalances in the pH of the environment in the brain is going to throw off neuronal activity. So that's all that means. So be familiar with what factors would influence cerebral blood flow. Let's see. Regulation of cerebral blood flow by oxygen. The big picture, what I want you to know from this slide is the fact that blood flow in individual segments of the brain can change as much as 100 to 150% within seconds in response to changes in local neuronal activity. What this statement is saying is that there are mechanisms in brain tissue which will allow for localized increases in blood flow depending upon which areas of the brain are most active, right? So if there's different areas, and this is carried out a lot by astrocytes. So anytime there are blood vessels in the brain, you will see astrocytes nearby. And the astrocytes will help, it's called neurovascular coupling, and it will help regulate blood flow to those areas. So the more active that neurons are in areas of the brain, astrocytes will help increase blood flow to those areas, right? Which makes sense. So if you have areas that are working a lot harder, these neurons are firing a lot more, they're going to require more oxygen, more glucose, so we need to get more blood to those areas. Okay, so just, it's the only statement that I want you to be aware of. So the concept of autoregulation with cerebral blood flow, autoregulation of cerebral blood flow is the ability of the brain to maintain really constant blood flow despite changes in perfusion pressure or despite changes in arterial blood pressure throughout the day, right? So you can have pretty vast changes in your blood pressure from moment to moment all throughout the day. But we want a very, very constant blood flow to our brain tissue. And so of all the organs in your body, the brain is the best at autoregulation. And so so cerebral blood flow, that autoregulation protects the brain from fluctuations in arterial pressure. And if we look at this graph down here, all this is showing you is, so we can have, so any fluctuations in arterial blood pressure from 50 millimeters of mercury all the way to 150 millimeters of mercury, this is a flat line. So on this axis, this is cerebral blood flow. And what this means is that from 50 to 150 millimeters of mercury, our blood flow stays super constant. It is a flat line. And up here, what this is showing you is the fact that, if we have this is showing you whether the arterioles would be dilated or constricted. And so at lower blood pressure, arterial blood pressures, we have vasodilation. And as we increase in blood pressure, we have vasoconstriction. But we're maintaining a incredibly consistent cerebral blood flow across a very wide range of blood pressures. So what do you need to know is be able to kind of like a definition type thing is autoregulation of cerebral blood flow is the ability of the brain to maintain very constant blood flow despite changes in arterial pressure from moment to moment throughout the day. Because delivering oxygen, delivering nutrients like glucose to the brain tissue is so incredibly important. We want blood flow to be incredibly constant. All right, so stroke, I'm sure everyone has heard of what ischemic stroke or hemorrhagic stroke is. Very superficial, what you need to know is ischemic stroke, which are going to be the majority, the most common types of stroke present, and that's going to be due to blockage of a major arterial blood supply to the brain. And this could be from plaque buildup, or it could be from a blood clot, something like that. But ischemic stroke is where you get blockage of an artery, and then downstream of that blockage, we're not able to deliver oxygen to the tissue anymore, and you get ischemia or lack of oxygen, and you can get death of that tissue downstream. Whereas hemorrhagic stroke is due to blood vessel rupture, and you get bleeding of the brain tissue. And if you get a buildup of blood within the brain, so everyone knows that the brain is comprised, or it's held in a cranium, and bone, there's not a lot of give to bone. And so if you have a buildup of blood, you're going to put a lot of pressure on tissue, and you can get death of tissue that way. So just be familiar with what ischemic versus hemorrhagic stroke is. And then as far as the outcomes of stroke, that's going to be determined primarily by which areas of the brain are affected. Okay. And then don't worry about this slide. It's just letting you know that the sympathetic nervous system and its ability to help control cerebral blood flow helps minimize the risk for stroke. All right, so let's finish up with CSF. So CSF, or cerebral spinal fluid, you should be familiar with what the functions of CSF is. So CSF is produced primarily by highly specialized capillary beds in the ventricles of the brain, primarily in the ventricles of the brain, secreted by the ventricles of the brain. And the functions are, it provides like a hydromechanical protection. Think of, that's a fancy word for it, it acts as a shock absorber. So we have very delicate brain tissue or central nervous tissue, right, in our cranium and our spinal cord, and it's encased in bone. And I mean, I'm pretty clumsy, so you walk around the day, you could your brain, you don't want your brain and spinal cord mashing against hard bone, right? So the fact that your CNS is essentially floating in a fluid provides hydromechanical protection in the form of like a shock absorber. CSF, also the fact that your CNS is essentially floating in CSF, it helps the CNS to become buoyant, and it reduces the effective weight of the CNS. CSF provides a way to transport nutrients throughout the CNS, and it helps remove metabolic waste products as well. So what you need to know from this slide is the fact that, all right, so here, these are the large ventricles contained within they're located kind of in the center of the brain, I guess is the best way to say that. And inside of these ventricles, you have specialized tufts of capillaries called choroid plexes, and that is what secretes the CSF into the ventricles. And then eventually the CSF can enter into the central canal of the spinal cord, as well as into the subarachnoid space, which surrounds the brain and spinal cord. And so now you can kind of visualize that your entire CNS is essentially floating in this CSF. CSF is constantly being produced and secreted throughout the day, and it's also being drained and removed. So there's this constant flow of CSF throughout the day, and that's where the fact that it's constantly being drained and renewed, new CSF is made, that's what provides that metabolic waste removal function. The only thing from this slide that you should be aware of is that the production of CSF is primarily driven by active transport of sodium ions, and you see this in a lot of tissues. The movement of sodium allows for the movement of other ions as well as water, but I wouldn't worry about this slide too much. Let's see. Okay, so from this slide, the only concept that I want you to be aware of is the fact that, okay, so the rate of formation of CSF remains constant throughout the day. So we have 500 milliliters of CSF being produced every day, and that's, as it's being made, it's also being removed. Well, how is it removed? So remember, CSF is made in those specialized tufts of capillaries called choroid plexes, located primarily in the ventricles of the brain. And that CSF, once produced, will circulate throughout the ventricles and then into the central canal of the spinal cord, as well as circulate through the subarachnoid space around the CNS. So here's the subarachnoid space, and then there are these protrusions from the subarachnoid space into these dural sinuses, and these are called arachnoid villi or arachnoid granulations. You can call them either one. But what's happening here is that old CSF will then get absorbed through these arachnoid villi into the dural sinuses, and the dural sinuses are part of the venous blood flow. And so this old CSF gets dumped into the venous blood flow, and that's how we get rid of it. So that's how all of the metabolic waste are removed from the CNS. So we don't get a waste buildup or a fluid buildup in the brain, right? So that's how it gets absorbed and it gets dumped into the venous blood flow. And being able to properly remove CSF as we're producing more throughout the day, it is really important because, again, if our brain is encased in a hard cranium and if we have any buildup of fluid, there's not a lot of give, and therefore where you can put a lot of pressure on brain tissue, and our brain tissue does not like excess pressure. So any sort of blockage in the flow of CSF could result in something called hydrocephalus. Now, babies are able to deal with hydrocephalus a lot better than adults are, and that's because their cranial bones are not completely fused yet, and so they can expand a little bit and deal with that extra pressure. But in adults, hydrocephalus is incredibly bad. Don't worry about this slide. I won't ask any questions. And then let's see, I think, all right, so what's the concept of a blood-brain barrier? The blood-brain barrier is made up of, there's three major components of the blood-brain barrier. It's going to be the endothelial cells of the capillaries, okay? The endothelial cells, astrocytes, which are neuroglial supportive cells, and paracytes. And it's these three primary structures that make up the blood-brain barrier. And for you, I just want you to be aware of what the major function of the blood-brain barrier is. And it's to very, very tightly regulate the movement of ions, molecules, and cells between, so to tightly regulate the movement of ions and molecules through the cells of the blood-brain barrier. Let's see. And so the blood-brain barrier, highly permeable to water, things like water, carbon dioxide, oxygen, and some of the lipid-soluble substances, but it's almost totally impermeable to plasma proteins and cells. All right. Any disruption to the blood-brain barrier could be really bad because maintaining the environment around the brain it's very delicate, and we want it to be very, very tightly regulated. So any blood-brain barrier disruption could potentially be very bad. So we don't want that to happen. Don't stress about this slide. Any form of edema or fluid buildup in the brain or the CNS is going to be really bad. And I've said this in our previous slides, is the fact that the brain is encased in that solid cranial vault. And so any sort of buildup of pressure because of fluid buildup is going to be very bad. It's going to affect cerebral blood flow as well as the function of the neurons. As far as brain metabolism goes, just know that the brain is not capable of anaerobic metabolism, and brain tissue neurons especially have a very high metabolic rate, so they rely heavily on a constant delivery of oxygen and glucose. And that's why maintaining cerebral blood flow is so important, because of the high metabolic rates of neurons. They need constant oxygen and glucose delivery. And that is it for my lectures. So just as far as this last video, anytime I said, like, this is the big picture concept for this slide, that's what you primarily need to know. So if you have any questions, shoot me an email, or I will be able to answer them whenever we do our review.